AS Level Biology 9700, Paper 33, May June 2024, question number 1. Yeast cells contain enzymes that catalyze the breakdown of sugars such as glucose and sugar and sucrose to produce ethanol and carbon dioxide. As the sugars are broken down, the yeast cells in a suspension will sink slowly to the bottom of the container, forming a sediment with a clear liquid above. This process is called sedimentation. You will investigate the effect of different concentrations of ethanol on the sediment of yeast cells. Test tube R, capital R in figure 1.1 shows how a test tube will be set up at the start of the investigation. Decide what you expect the contents of the test tube to look like after sedimentation on test tube S in the figure 1.1. Draw line to show the layers that you expect to see after sedimentation, a label line and label to identify the sediment. So sedimentation, the layers, it will be, you can say the level will be the same as in R and sediment. Now, next is you are provided with the materials shown in table 1.1. Label E 100% ethanol, A calcium chloride solution, W is distilled water, and Y is the dried yeast. Students, you know, this is the experiment which has to be done in the lab. So I will guide you in this regard how you will perform in the laboratory. So if any solution comes into contact with your skin, wash off immediately with cold water. You should wear suitable eye protection because you are using the chemicals. So you are going to wear the goggles. You will investigate the effect of different concentrations of ethanol on the sedimentation of yeast cells. You will need it to prepare different concentrations of ethanol. Measure the height of the yeast cell. Yeast sedimentation in each concentration of ethanol after 4 minutes. You will use proportional dilution to make different concentrations of ethanol. You will prepare 10 cubic centimeter of each concentration using E and W. Table 1.2 shows how to prepare two of the concentrations you will use. Decide which other concentrations of ethanol you will use. So here complete table 1.2 to show how you will prepare the concentration of ethanol you will show. So here you can see, students, the table 1.2, they have given two concentrations, that is the 100 and 60. And percentage concentration of ethanol is 100, in which the volume of ethanol is 10 cubic centimeter, whereas the volume of water is, they have not added the water into it, they have used the pure 100% ethanol in the first. And the last concentration which they have labeled is 60, in which the volume of the ethanol has been used is 6 cubic centimeter, whereas the volume of water has been used is the 4. So we have to make 3 more concentrations in this, which should be between 100 and 60. So below 100, we can use 90, in which 9 cubic centimeter of ethanol and 1 cubic centimeter of water will be used. And the other concentration which we can use, that is 80, and in which the 8 cubic centimeter of ethanol and 2 cubic centimeter of water. And the third one which we are going, that is the 70%, and in 70% there should be the 7 cubic centimeter of ethanol and 3 cubic centimeter should be the use of water to make solution of 10 cubic centimeter of having these concentrations, that is 100, 90, 80, and 70. So in the guidelines which they have provided us by making 100% ethanol and 60% concentration of ethanol, we can make these three different concentrations in between these already given 100 and 60. Now we will move further. Carry out step 1 to step 15. So you have to perform these steps which are 1 to 15. So what we are going to do this? We are going to do this. In these beakers provided, prepare the concentrations of ethanol as shown in table 1.2. We will prepare and the label one test tube with the C. One test tube with C. So we will one test tube. It is C. Label the other test tubes with the concentrations of ethanol prepared in step 1 and other concentrations. They are, first of all, we can draw over here for your convenience, students. This is for 100. And all 5. We will label them as 100. It is 90, 80, 70, and 60. On one side, we have labeled with C. Now, in the beakers, I have written on the side of the paper as it was not moved, then I will move the paper. So, label one test tube with the label C. We have labeled C. So, this is the control. C stands for control. Label the other test tubes with the concentrations of ethanol prepared in step 1 as 100, 90, 80, 70, and 60. So put 1 cubic centimeter of A into each of the test tube labeled in step 2 and 3. So what is A? Already it was given, that is A is the calcium chloride solution. So we will add 1 cubic centimeter of ethanol into each of the test tube labeled in 2 and 3. 2 and 3 means in control test tube, and the other 5 test tubes with concentrations 100, 90, 80, 70, and 60. So first of all, I will add into the control, that is 1 cubic centimeter of A in all of these. 1 cubic centimeter A. I'm writing for your convenience to understand. It will help you to understand what we have added into these test tubes. And when you will be in laboratory, it will be easy for you to follow. Then put 7 cubic centimeter of W into the test tube labeled C. 7 cubic centimeter W. What is W? W is the distilled water. So first of all, in all test tubes, including control, we have added 1 cubic centimeter of calcium chloride solution, and then we have added 7 cubic centimeter of water into the labeled C. Then in the next, put 7 cubic centimeter of 100% ethanol E into the test tubes labeled 100, 7 W ethanol. Now we are using ethanol. We have added water into the, and then in all of these. In control, we have added water, but in the other concentrations, we have added ethanol. We have not added ethanol into the control experiment. Put uh, Now put 100 cubic, repeat 6 for each of the other concentrations prepared in step 1. We have done with all. Now put 100 centimeter cubic centimeter of W. W is the distilled water into the beaker labeled Y. Uh, this is the beaker. It was labeled Y. And we have to add the 100 cubic centimeter of water because Y was the dried yeast, and we have to make a solution of the dried yeast by adding 100 cubic centimeter of water into it. Now we have 100 cubic centimeter of water, dried yeast solution. Now it is dissolved into it. Then we will repeat step 9. Step 9 for each of the other labeled test tubes, including we will add, stir the yeast cell suspension. We will dissolve it by stirring and then put 7 cubic centimeter of the yeast cell suspension into the test tube labeled 100%. So labeled 100%, we will add 7 cubic centimeter of yeast solution suspension. Same, it will be on. It's very difficult to write on screen. And we will also add it into yeast suspension into the control. So what we have done, first of all, we have made concentrations. Then we have taken one test tube. With control, we have added 1 cubic centimeter of calcium chloride solution in all of these test tubes, including control test tube. Then we have added 7 cubic centimeter of ethanol into the 5 concentrations test tubes with 5 concentration 100, 90, 80, 70, and 60. But in control, we have added 7 cubic centimeter of water, distilled water. And then we have made a solution of dried yeast by adding 100 cubic centimeter of water into it. Then in this way, we used this yeast suspension. Then we added 7 cubic centimeter of yeast suspension in all of the test tubes, including the control. Now put a clean bung into each of the test tube and invert the test tube to mix the contents. So what we will do, we will make use of a bung. In this way, I will draw only on one test tube. We will close it and then we will invert it and we will shake it thoroughly as the suspension or the solution, all these solutions which we have added should be mixed together. So repeat step 11 for all the other test tubes. We will repeat this for all of these test tubes and including this. Then start timing. Immediately measure the height of the sediment in each test tubes. These are the results at the start. 0 minute. record your results in table. We will make a table for this. After 4 minutes, after every 4 minutes, we have to take the height. What is happening in these test tubes, including the control, we will note down the height of the sedimentation. After every 4 minutes, we have to measure. So repeat step 14 so that the results are recorded every 4 minutes until the final results are recorded at 20 minutes. Record your results in an appropriate table. And here in this case, students, there is one independent variable and one is dependent variable. So the independent variable in this case is the concentrations of this ethanol, and the height of the sediment formation is the dependent variable. So we will make a table. Yes, we are going to make a table in the space provided in this way. We will need to in this way, we will make headings. So in this way, we will make a table, and on the table, we can, on one side, we will write the time 0, 4, 8, 0, 4, 8, 12, 16, and 20. And here we will make use of the concentrations and it will be 100 over here, 100, 90, 80, 70, 60, and control. Then height, in millimeters. Height in millimeters. Now, students, we have to measure the sedimentation which has been formed in these test tubes. First of all, when we uh, shake it at 0 minute, then we will take reading over there. What will be the reading? At that time, you will note it down over here. Then after 4 minutes again, you will note down the height of the sedimentation. And then in the after 8 minutes, and after next 4 minutes, which will be 12 minutes, and then after next 4 minutes, 16 minutes, or 16 minutes. So in this way, you will have to measure the height by with the help of scale, and then you will write it down over here, and in this way you will be able to get the 5 marks if you are measuring the correct heights. In this way you can make the table. So it is height in millimeter, which I have written. I have tried my best always, as always, because I always write on the screen. Now, after doing this, we are moving after we have got these results. When we will do this experiment in the laboratory, then we will got this. Now, using your results in A, state one conclusion that you can be made about the effect of the concentrations of ethanol on the sedimentation of the yeast cells. Now, students, here you have to state the conclusion. The which conclusion? After making a table, after taking a height, you will be able to state a conclusion. After taking the reading, you will measure it with the help of scale. Then after taking that conclusion, then you will be able to write over here. So your observation, measure the height according to the instructions, and write your conclusion about the concentration of ethanol on the sedimentation of yeast cell. It means you will observe which concentration has more effect on the formation of sedimentation. That is, it is highest at 100 or 90 or 80 or 70 or 60. You will observe that, then you will write it down over here. Then the next question is, a student suggested the hypothesis ethanol is needed for sedimentation of yeast cells to occur. It means after these results, he will be able to follow this hypothesis. So using your results in A3, state whether you support or reject this hypothesis, explain how your results provide evidence for this decision, support or reject. So you have to support or you have to reject. It will depend. It will depend. It will depend on the results which you have obtained after doing all those steps which we have did previously, step number 5 to step number 15. So after measuring the height, after making a table, you will be able to reject, or you will be able to reject or accept this hypothesis. So whether you are rejecting or accepting, it will depends. It depends on the results you observe at the time in the lab. At that time at the lab. So you will reject, you will support. It depends on you or it depends on your results. Now we are moving towards the next question, because this is experimental work. So I can guide you only, but your results uh, depends on the investigation which you will perform in the laboratory. So using the results at 20 minutes, state this concentration or concentration of ethanol that caused the most sedimentation. So here you are going to write which one of them, that is out of 100, 80, 90, 80, 70, 60, which of these you have got the most sedimentation, and then suggest how you could modify this production to obtain a more accurate estimate of the concentration that causes the most sedimentation. How yet you can improve to get more sedimentation. So here you can give the idea, because first of all you need the results. From the results you will come to know in which concentration uh, the sedimentation was high. So according to those results you can uh, make better decision, or you can modify your results. As for example, I want to give one example according to your marking scheme, that you can use the concentration of ethanol either side of concentration stated, or you can use the narrow intervals close to the concentration that cause the most sedimentation. As for example, you can observe after 2 minutes time can be reduced, time can be reduced, that is after every 2 minutes, every 2 minutes, or concentration range can be changed, can be reduced, you can say. Instead it can be between the 100 to, it can be less than 100 and above than 90, or it can be 95%. So the other concentrations can be used. Other concentrations of narrow range can be used. So these are the ideas which we can give you. Again and again, I'm saying to you that I am giving you the ideas, but everything will be according to your performance in the lab. Identify the main source of error in the investigation into the effect of different concentrations of ethanol or on the sedimentation of yeast cells. Yes. Here you can see that uh, a source of error. Source of error, that is when you are measuring the time, you have not measured the time properly, or you can say when you are measuring the time, there can be the change, as for example, uh, change in this way, that is the length of the time was different for different considerations. It can happen, and when you are starting your stopwatch at that time, the can also affect the, that is for one concentration you have taken more time as comparison to other. So test tubes were not left for the same length of time. I have given more ideas. Test tubes were not not left for same length of time. While you were measuring the time at that time. During measurement, you can make mistakes. Another possible source of error when carrying out step 6 of the investigation is shown in table 1.3. Complete table 1.3 by stating whether the type of error is systematic or random, and the effect the error may have on the trend seen in the results. So the 7 cubic centimeter of mark on the syringe used to measure the volume of ethanol actually measured 7.1 centimeter. So this type of error, you can say it is systematic error, because when you are measuring with the help of syringe, uh, quite possible, when you are pulling the plunge, then at that time you pull more than the 7. It can be 7.1, so, or it can be less than 7. Sometimes it can happen that you are, and you are not taking care of it. So in this way, you have to take the more or less, but it is systematic. We will write systematic, that is, you have not used the syringe properly. So effect on the trend, so it will not affect the trend because a very small quantity is greater as comparison to other test tubes. So it will not affect on the trend because it is very small quantity which you have used, which you have taken. Okay, next is the part of this question, which is in fermentation, the action of yeast converts some of the carbohydrates in plants to ethanol and carbon dioxide. A scientist investigated the production of ethanol during the fermentation of carbohydrates from different sources. The investigation was carried out at 30 degrees centigrade and at pH 5. Using the yeast, saccharo, Saccharomyces and cerevisiae, all other variables were kept constant. Source of carbohydrates, molasses, 3.80 percentage ethanol produced per 100 gram of carbohydrate, and in oranges it was 1.05, and grapes 2.65, and in beetroot 3.35, rice 4.70. Now question is, draw a bar chart on the table 1.4 on the grid in figure 1.2. Use a sharp pencil. So we are going to make a graph over here. So on graph, when you are making graph, so you on x-axis, you are going to take the concentrations of carbohydrates, and on the y-axis you will take the concentrations. You will write the name over here. Sources of carbohydrates. Sorry, sources of carbohydrates, and it is 0, and then after 10 it is 1, after 10 it is 2, then after 10 it is 3 and 4, and this is 5. So on y-axis you are going to take the concentration of ethanol. That is, first of all, again it will be 1, it will be 2, sorry, it will be 3, 4. I am taking it very roughly because I'm just giving you the idea that how you are going to draw it. Now, for drawing, students, first of all, uh, you have to label x-axis and y-axis. Then you have to draw the bars. Bars should be of same width, and they should not be joined together because this is the bar graph. And then secondly, their width should be same, their height should be properly, uh, you can say line should be drawn with the help of scale, and then the distance between the bars should be the same. And now I'm going to draw the first is 3.8. What molasses? It is molasses. And second, oranges. And our third is grapes. And fourth is beetroot. Beetroot. Sorry, space is very less. So, and at 5 is rice. 10 small scales should be equal to 1. But I have not counted properly. So you will count properly. You have to take one small scale should be equal to 1. So in lessons, first of all, 3.80. So 3.80 is nearly equal to this. It is 1. It is here. So I will draw the bar. I will draw between 4, 4 and 5. On screen, I am unable to draw with scale. So one should be in the center. In this. And then the second is 1.05. Orange is 1.05. 1.05. So it is maybe equal to in this. Little smaller than 1. It is little bit difficult for me to draw properly, but I am trying my best to draw them. In this way. But please, you have to draw very carefully. You have to got the marks against them. So be careful while you're drawing. The bits should be the same. I have not drawn just like that. Then grapes 2.65. I'm just giving you the idea. I'm not perfect at this time. But I'm giving you the idea. And then the third 3.35. 3.35 at 4, it is near equal to in this way. And then 4.70 nearly equal to 5. It is over here. I have drawn so many lines, but there should be a very clean drawing. Students, you are perfectionist. I'm not just like that at that time. So in this way, you can draw your graph, bar graph. You have to draw it with pencil properly. It should be drawn at that time in the question paper, in the examination hall, in the lab. But I'm not perfect as just like you. So be careful while you draw this. Now next is the investigation was repeated using a different yeast Schizosaccharomyces probe. All the variables were uh, kept in the same as in the first investigation. The percentage of ethanol produced using Schizosaccharomyces probe was found to be lower with all of the sources of carbohydrates. Results are shown in table 1.5. You can see, and then uh, you can see the results are a little bit different. The molasses 3.8, but in the other Species was 3.68, oranges were 1.05, they are 1. Grapes were 2.65, now they are 2.57. Beetroot was 3.35, now it is 3.29. Rice was 4.70, now it is 4.34. Now the question is, suggest why the percentage of ethanol produced by uh, Schizocaramyces from lower than the percentage of ethanol produced by Schizomyces cerevisiae for all sources of carbohydrates. So why it is different? And the S cerevisiae and the S form, the different types of sources of carbohydrates. So quite possible they work at different, you can say, different situations or conditions of temperature and pH. So quite possible that is the due to the changed pH or changed temperature, they have not given the same results. So we to know that the every uh, different types of uh, these carbohydrates, they you can say concentration is changed due to, you can say that quite possible the due to changed pH or temperature, the results are different. Plus the different sources of carbohydrates need different range of pH or temperature, due to which the fermentation gave different results. Different fermentation gave different results. It happens, it is fact, that is, they do not work in the same condition. They need different conditions, they need different pH, they need different temperature. So in this way, the ethanol which was produced by using these two different varieties, it was different. So it was all about question number 1. Please students, like, share and subscribe my channel. Thanks for watching.